Now when it comes to the number of the nucleotides present, there are 100, almost 100 nucleotides. Remember, these numbers are round off figures. It can be few numbers up and down always possible. So there are 100, almost 100 nucleotides present in the 5S and 5.8S type of rRNA, and it is 1500 or somewhere close to 1500 number of nucleotides present in 16S and 18S type of rRNA, and 3100, it can go even up to 4000 also. 3100 near about plus and minus is possible, would be the number of nucleotides possible in the 23S and 28S rRNA. So there are various types of rRNA possible, and they are variable in their properties also. And then when we all know, if you add more number of nucleotides, then the molecule will become heavier. We all know about the molecular weight concept. So accordingly, the rRNAs are classified into three categories: high molecular weight, then again high molecular weight but comparatively lesser, because here the number of nucleotides are almost half, and low molecular weight type of, some books will mention it as the moderate molecular weight. So high molecular weight, moderate molecular weight, and low molecular weight types of rRNA. So these are the values which tell you the molecular weights: 35,000, 550,000, and then 1100 to 1000. So these are the values of the molecular weights of the various types of rRNA possible. Now just remember that rRNA are the longest of all the RNAs, because when you pay attention to the number of nucleotides present in the 28S or 23S of eukaryote and prokaryote respectively, they carry a huge number of nucleotide, which generally ranges between 3000 to 4000. So this is a long number of nucleotides. That's why they are the longest of all the rRNAs. Remember, rest of them are small. And we all know that rRNA will give rise to ribosome, and ribosome is a structural component of the cell. That means that ribosomes are going to be stable, and they are going to be for a long time inside a cell, whether prokaryotic cell or eukaryotic cell. Therefore, rRNA are the most stable form of RNA. So this is the most stable form of RNA, and remember, there are very less modifications present in the rRNA. But modifications basically means the nitrogen bases undergo some side group changes, some modification in their structure, and that modification is called as the modifications of the rRNA. Then remember, because it's a long chain, so it is impossible for this chain to remain uncoiled. So there will be some portions in this chain which will be uncoiled, but some portions will definitely undergo coiling. And remember one thing, whenever it is uncoiled, those areas will have unpaired bases. Remember, in 11th we have done, whenever A will face T, it will develop 2 hydrogen bond. Whenever G will face C, it will develop 3 hydrogen bonds. So that same concept is there. As long as the thread remains long and AGTC are arranged like this, they will not develop bonds. But whenever bending takes place or coiling takes place, when 2 complementary bases face each other, that means A facing T or G facing C, they start developing bonds, and those bonds are mentioned here as paired bases. That means this long chain will not remain like a linear chain. It undergoes coiling, and this coiling area will develop paired bases, so stability would be given by it. Remember, rRNA forms the ribosome, and the formation. So when we talk about zygote and embryogenesis topic of 12th class, we have already done that. The rRNA formation starts during gestulation. So formation of rRNA starts quite late when you compare it with the tRNA and compare it with the mRNA. rRNA formation starts very late, and it starts during the process of gestulation. Now the enzyme which is involved in the case of eukaryotic machinery which causes the formation of rRNA is RNA polymerase type 1. So this enzyme is responsible for formation of RNA from DNA. And remember, when rRNA is formed, for example, in the case of eukaryotes, we have learned how many types are there: 28S, 18S, 5S and 5.8S. Now 5S is always formed separately, but whenever 28S, 18S and 5.8S type of rRNAs have to be formed in the eukaryotic machinery, they are formed from the same primary transcript, which is 45S in length. That means when DNA forms rRNA, it will first of all form a longer piece of rRNA which is 45S in its nature. Then it undergoes cleavage. Now if you remember, I have already told you cleavage is the concept. See, I have mentioned it here already. There are enzymes which are catalytic in nature and they are able to cause cleavage. So ribosome is one of the enzyme which causes cleavage. So what exactly is cleavage? A long piece of like 45S long piece of the rRNA undergoes cutting, 2 times cutting, and this cutting will create 18S, 5.8S and 28S type of rRNA, and this give rise to the formation of these 3 types of rRNA which are present in the larger and the smaller subunit of the eukaryotic ribosome. Remember, during this process, extra RNAs are there which are removed and digested, and during this process modification occur by the process of methylation. Remember, just like DNA, RNA also will have 5' to 3' ending. So in the DNA topic I will explain this. I don't want to repeat the topic again. So in DNA I will explain to you how exactly 5' and 3' are mentioned.